Hi, and welcome to this quick video on how to use diagnostic ion data analysis reduction for single cell proteomics quality control. I'm Ben Osborne, and I'm just going to walk you through this. Um, DIDAR QC is a short and simple, lightweight Python script for performing quality control measurements of multiplex single cell proteomics data. This is originally developed by Connor Jenkins for finding post-translational modifications of reduced diagnostic fragment ions, and we've adjusted this for multiplex single cell proteomics. And you see in this little cartoon on the left, what we're throwing to trash here is a scope MS spectra where we have a nice high carrier channel and it's fine. We've got, our red mark here is our method blind control. And here we only have a signal from a couple of single cells. And so we trash that spectra. And what we are doing is keeping the spectra that look great, where we've got reporter ion signal from all of the single cells or some user defined threshold for the single cells. And so, it's taking a step backwards if you think about how scope MS or multiplex single cell proteomics works. We've got a method blind control or two, and we have single cells that are just labeled with reporter ions, commercial reporter ion tagging reagents, and we have a carrier channel. We pull them all. We're using a timsTOF using passive RIQ or reporter ion quantitation. And what we get is our signals for our reporter ions, and we get fragment ions that help us sequence our peptide. All we've got to identify that the peptide from the single cell is there and its relative quantity is just this one single mass. So the idea is here, hey, what happens if we've got a cell that fails technically? It's really, really hard to prep a single cell. Static electricity can knock them out of plates. You can have them stick to the side of the well. You can't see it. It's very easy to lose one cell or a lot of cells. And so what DIDAR does is it does a couple of things. It finds spectra that contain fragment ions that you give it. And it is based on the original DIDAR script that Connor wrote during his master's work at Hood College. But for DIDAR, we find spectra that contain fragment ion masses you give it. Spectra that contain those fragment ions are made into a new file. So in this case, I'm giving the diagnostic ion is the reporter ion exact mass. And then we give it a user specified mass accuracy tolerance. And then it also generates a diagnostic test that provides metrics for each file. And that's what we're really going to use for QC here. And in the context of scope MS or multiplex single cell proteomics, I'm using these diagnostic files, laying them out, and you can see here's an example across. These are files that we've acquired on the timsTOF, and this is the permanent suffix that the instrument assigns to files. And we see here in file 775, something went wrong with cell number 120. It was labeled 127 in this run. Right, where all the rest of them, maybe the 128 wasn't too great either, but here we've got 6,000, for spectra with reporter ions from 131, but only 600 from 127. So this is a flag for, hey, something went wrong with that cell. And in addition, we can flag entire runs where something went wrong. This entire LCMS ejection 782, basically no reporter ions across the board from single cells, maybe something from a 128. And then here I've highlighted just, you really can see the upper outliers too, and cells that went really well. So DIDAR can give you these really fast metrics and these little text files that it generates that they can tell you as you're going through optimization, what you're doing right, what you're doing wrong. And we found this really helpful as we could just generate this text file really fast to know if that last run was okay and if we should try that again during optimization. So we can also take these and here we just take these diagnostic outputs and graph them in things like GraphPad. And here you can see those same two failures. The 775, 128N was bad, and so was the 128N and 782. But in 782, it was all of them, right? And so that's basically it. Except for we can go from we can look at intra-batch, intra-batch variability. Here we've taken all of the single cells and the number of spectra that they contain reporter ions from the scope 2 data set from 25 or 50 files randomly chosen from that. And we can kind of see the intra-batch, interbatch variability and other, just useful metrics from this. But I want to pop over to just show you how to use that, because that's what this video is for. I'm not going to open Perseus. What we're going to need is the Anaconda Navigator. You can Google this if you don't have it on your desktop. Yeah, you can get it. It's easy to find. And there are other ways, there are other IDEs out here. There's other ways to run Python scripts. This is just the way that I'm doing it because it's straightforward. And then once you get Anaconda Navigator, Spider is a scientific IDE. It's already pre-installed. It makes a lot of sense to use it here. And then you would first install it. This orange here is not installed on this computer, or at least in this instance of this Conda. But Spider is, and I can launch that, and launching it would bring me into here. So what you're going to need from for DIDAR is you're going to go to the DIDAR SPC/QC GitHub, and you're going to download some files. And what you're going to need is just to unzip that file into where your data is. And so when you unzip that DIDAR SPC/QC v1.0, you're going to get a diagnostic ions text file, the DIDAR Python script, and an extra glycoreporters text if you're interested in going after reporters for glycans, and I am. So here we're going to use those as well. But the diagnostic ions text, when you open that, it's really simple. It's just got the exact masses of the TMT reporter ions that were used in this study, and you can just copy those from wherever you get exact masses for diagnostic or for reporter ions. And then you want to copy. It's a really tiny. This is kilobase, right? This right? So you can copy this anywhere. What we want to do is put this in the file where your MGFs are that were generated from your scope MS data, and open DIDAR.py, this instance in Anaconda, which I've done here, and you get this little script. And again, this is extremely lightweight. It's only a couple of KBs here. And what you're really interested in here is the mass tolerance for your diagnostic ions from your text file. And here we're doing timsTOF data. It's been calibrated with the passive RIQ calibrator, and we're still doing 0.0005. For Orbitrap data, you might get to do 0.002, but these are the settings that we use. And then the other thing that you want to check here is how many of those diagnostic ions do you require that you have to keep this? And for illustration purposes here, I'm just going to go ahead and say, like, if you have one diagnostic ion from one single cell, I want that data. So I'm just going to keep that one. But if let's say, hey, like, if I will only be interested in a spectra if it's got a detectable reporter ion with it from at least 5 single cells, then I just change this number to 5. Right? 5 or more single cells, and that's all we do. And then you hit the play button. What'll happen here is a whole lot of text will start running, and as it's going through this timsTOF file, you get the scan header for it, and it builds through. So I'm just going to skip ahead. I already processed this earlier. We'll go to processed. I'm going to show what the diagnostic text looks like. Wrong one. Wait. Oh no, that is the right one. Okay, so what we've got here, and this is how lightweight this is, from this file, there were 89,959 MS2 spectra that contain the 126 reporter ion, 126,000 that had the 127. And this is from 8 single cancer cells that were loaded onto timsTOF SCP as an analysis. And 132 was our method blank in this. So there's basically no reporter ion in the 132. It's random shot noise and everything. You're going to see some 132s. I'm still looking at the glycos, and there's a surprising number of glycan reporters, but I gave a pretty large library of diagnostic ions. But in general, we get about 108,000, somewhere between 70 and what's the highest number, 126,000 MS2 spectra that contain reporter ions from these single cells, and that's what I get from data. And then if we look at the MGF, so for something like the timsTOF SCP, when you run through a reduction like this, you don't expect a great big drop in the number of in the data density, because you're really not throwing out that many spectra unless something went really wrong. These cells were prepped by a robot and were then and then were analyzed at a really low flow rate. There are a few missing channels compared to some of the other data, the ones I showed in the PowerPoint. But really, that's it. This is the MGF. You can take that cleaned up MGF and then just search it in any of your normal pipelines for the downstream analysis. But that is all I've got. Thanks for checking out this video.